How do mutations arise in our cells? So mutations exist in two flavors. There can be those that are introduced from an outside factor or those that are introduced from an inside factor. So if the mutation is coming from an outside of the cell, so something outside of the cell is causing it, it's called an induced mutation. If something inside of the cell, or like a cellular process is causing it, it's called a spontaneous mutation. Okay? Now, within those two, spontaneous and induced, there are still a lot of things that fall under it. So here you see a host of ways that we can introduce spontaneous mutations. And then we're also going to talk about, even though we only list two here, chemical and physical for an induced mutation, there's still different ways in which it can occur. So spontaneous mutations. Mutations that arise from internal factors. Something within the cell is causing a mutation in the cell. The first one we'll talk about is depurination. Okay, so remember our purines and our pyrimidines? Well, we can have a change that causes us to remove a purine. Okay, so here we have a DNA sequence and we have a removal of a G. So that introduces this apurinic site. So the removal, a lack of a purine in the site. So now we have this gap in the site. So what happens? Our DNA repair mechanisms come along and they try to fix it. But it's two things that can happen. One, they fix it correctly, all is well. Two, they don't. So remember that each of these serves as a template, both strands of our DNA. One strand is fully intact. That C is there where it belongs. You put the C there, you complementary to it, it's all good. Well, the other one is missing. So now we're going to play guess what the sequence was. And if you don't get it right, you've introduced a mutation successfully. We have deamination. So in amination, the amine group of a cytosine comes off. So when we take that amine group from that cytosine off, we no longer have cytosine. Now we have a uracil. So now what happens with that uracil? Well, hopefully our repair mechanism will recognize, hey, this don't belong here and put the correct thing in. But if not, when we split this uracil is going to bind to what? An adenine. So where it was a CG, we now have a UA. Hopefully that adenine is replaced. If not, we have a UA. If it is, we might have a TA. But either way, it's not the same sequence that it once was. We have these tautomeric shifts. So think back to chemistry and tautomers. And this is where we know like molecules are always undergoing these changes. And so it can exist in two forms here, the keto and the eno. And now depending on where it is, it could look like something different. Okay? So here we look at two shifts and how they're going to bind. If we have these tautomeric shifts, thymine can bind with guanine and cytosine can bind with adenine. Now we are violating Chargaff's rules. And if we look at this, so we have a thymine and an adenine and we have a shift. Well, now we have a base pair change, a T to G, and we keep our T to A. Well, here again we split it. This T will bind with the A. This G is going to bind with the C, and now we have a mutation. In our cells, we have things called reactive oxygen species. So these tend to be like charged molecules that can then go on and attack things within the cell. This is a natural byproduct of some reactions like making ATP, so oxidative phosphorylation can produce oxidative stress in the form of these reactive oxygen species. Our body has ways to fix this, to get rid of it. But in the event that we can't, well, now we have these things going around wreaking havoc in our cell. Okay, so here we can see we had a guanine, we oxidized it, it's going to make some changes to it. Okay. Our dear friend, those trinucleotide repeats are back again. So here You have your trinucleotide repeat agent, and our DNA is going along and it's putting it in there. Well, all these things are alike. And so now we get this hairpin in part of it that forms. And when this hairpin forms, our polymerase just goes on about its job. The hairpin separates because it shouldn't be there. Now we have a gap in the DNA. So this is how we elongate those trinucleotide repeats. So remember we said with each successive generation, it gets longer and longer. But this is how it gets longer and longer. Then there are some other things that can happen. We know that polymerase is capable of inserting mismatched base pairs. Although it's rare, it can happen. We looked at this a little bit with the trinucleotide repeats, but sometimes you get these strand slippage. Okay? So while we're trying to replicate, something happens, polymerase might lose its place, come back in, but we have the addition or subtraction of a base pair. So now we have our indels and those nucleotide expansions. You can have unequal crossing over. So something happens where our homologous chromosomes misalign. When they do that, it's an unequal crossing over. We can insert or delete things accordingly. Okay? And similar to the reactive oxygen species, there's something called reactive nitrogen species and things. So just normal metabolic pathways can give off these detrimental byproducts. And then we've talked about transposable elements. Depending on where they insert themselves, they might disrupt a gene's function. Okay? So those are all spontaneous. Now we're going to look at the induced. So these are ones that's coming from external factors. We usually call these mutagens. So any environmental agent that increases the rate of spontaneous mutation. So one way is these base modifications. So what happens here, and you'll see that some of these seem similar to things we just talked about. So you can put some molecules in, and they will replace amino groups with keto groups. When you do that, you can change the way things base pair. Okay, so if we can do that, we can create these mismatched base pairs, and similar as before, one strand might be accurate, the other won't, and now we're introducing a point mutation. You have these intercalating agents. So what they do is they go and put themselves inside the double helix. So think of them as being a similar size as the nitrogenous base pair. And so they will kick out a nitrogenous base pair and put themselves there. Now you're missing one. So when you try to fix this, you don't know what it is. So you just put in a nucleotide. You have base analogs. So these are compounds. So if you look here, this 5-bromouracil, these are compounds that look similar to our nitrogenous bases. And they come along and insert themselves. And depending on which one they're supposed to be, they can cause a change in our sequence. We have ionizing radiation. So these tend to be like our X-rays and gamma rays and things. If they penetrate, they can produce free radicals, similar to those oxidative species and things. These free radicals are going around causing a lot of havoc to our DNA. They can delete bases, oxidize them, introduce single-strand breaks, cause a cross-linking in our DNA, and even introduce chromosomal breaks. And then we have our non-ionizing radiation. This is like our UV. So UV is known for making these thymine dimers. So under normal circumstances, thymine binds to adenine that's across from it. Well, if it's a thymine dimer, you break those hydrogen bonds with adenine, and those two thymines who are next to each other want to bind. And this is caused by UV light. Okay? Now, the mutation rates. So we're always trying to figure out what's the likelihood of mutation. So the mutation rate is the likelihood that a gene will be altered. And you can look at this as being the number of mutations in a gene, in a cell, in a generation. Or you can also look at it in the non-coding regions, because we know those are more abundant than our gene coding regions, and they are also susceptible to mutations because essentially they are DNA sequences as well. And the rate of mutation is not stagnant, so it can change depending on the environment. So think about like if you hang out in the sun, You're exposed to UV more than someone who's not, so that may increase your mutation rate. Okay? How does that sound?